Good day, good day, good day, good day. Welcome to our second lesson. Last time we covered the DNA and also RNA. We were focusing on the differences. For example, we know that DNA has got deoxyribose sugar and RNA has got the ribose sugar. We also know that DNA has got thymine and then RNA has got uracil. And then we also know that DNA is coiled and also double helix or double stranded, whilst RNA is only single stranded. So those differences are very crucial because I've seen some questions in the past papers where they're asking them to tabulate differences between DNA and also RNA. Today I'm going to cover DNA replication and also DNA fingerprinting. So without wasting time, let's go straight to the topic of DNA replication. When I was still in grade 9 and 8, I remember my teacher used to always emphasize on us that if you're good at something, try to learn more about it. Try to pay more attention and learn more about that content, because the more you learn about something, even your knowledge always increases. So same thing, I want to say it applies to DNA replication, because I want to end up looking like maybe I'm saying that DNA wants to increase its knowledge, or no. Just want to give an example. During DNA replication, the DNA normally makes copies of itself. These copies are done in different rounds, which we're going to cover today. So this process is mostly controlled by the DNA polymerase. There are other enzymes which are also present. So if you guys want to do medical linked courses or life sciences, you learn more about those enzymes when you do first year or second year or third year level or postgrad. So I really apologize for my drawing. I'm not good with drawing. I really apologize. So here I was trying to show you how the process of DNA replication occurs. So for example, we know DNA is double helix. That's a double helix. It has got two strands. So this DNA that I'm having here is having two strands. This strand number 1, strand number 2. And we also know from last lesson, adenine always pairs with thymine, and they've got the two weak hydrogen bonds that will hold them together. Same thing, thymine will also pair with adenine, two weak hydrogen bonds will hold them together. We also know that guanine also pairs with cytosine, and the three weak hydrogen bonds will hold them together. So let's say I'm having this DNA molecule here, and then it undergoes DNA replication. So first thing first, the DNA polymerase, that enzyme, will break the weak hydrogen bonds. So now the bonds are no longer there to hold my adenine and also my thymine and also my guanine and also my cytosine. So they're broken down. The enzyme will break the weak hydrogen bonds. Now it becomes two strand. This strand here becomes by itself. Same thing as this strand. They will become by themselves because they are taken away from each other. So now technically I've got two different strands. So now there will be some free nucleotides which will be in a nucleotide pool or a nucleotide sep. Those nucleotides which are there, they're just moving around there, waiting to bind to something. So here you must be careful. This strand is this one, ATC. It's this one, ATC. So even this strand here is ATC. So the free nucleotides which are there moving around, there are different mixtures. It's A, this T, this G, and this C. And we know A will always complement with T. So the T that is moving around in the nucleotide, it will come and attach to my A. Same thing, the A that is moving around will come and attach to my T. Same thing, the G that is moving around will come and attach to my C. So now I've got a new DNA molecule. This is a new DNA molecule which was formed by this strand here. So this DNA molecule was formed by this strand. And again, I've got this other strand also by itself. So again, the free nucleotides which are moving around, we know A will always go to T. So the A that is on the nucleotide pool will come and attach to T. Same thing, C that is on the nucleotide pool will come and attach to my G. So now this strand here, it will form this other copy of DNA. So this first strand, it forms another copy of DNA. Same thing as this one, it forms another copy of DNA. So I've got two copies now. Originally I was having one copy. Now I'm having copy number 1 and then also copy number 2. So I'm having 2 different copies, this one and also that one. So that's the whole process on the concept of DNA replication. DNA was initially 1 copy, now it becomes 2 copies. So this is the first round of DNA replication. Initially, we've got 1 DNA molecule. After the first round, we've got 2 copies of DNA. So yeah, I just made a summary of the whole process. In case if maybe I was moving a bit fast, these people can just see on the slides the points that I was highlighting. So remember, the weak hydrogen bonds will break due to the polymerase, and then those 2 single strands will be there. And then the nucleotides which are there in the nucleotide pool or SEP will then bind to each strand. Then we end up forming complementary base pairing, which will lead to me forming a new copy of DNA molecule. So here, I apologize. I'm going to do some bit of math. So if I've got people here who don't like math, I apologize. I really apologize. This n is equals to 1 represent the first round. So I normally use this formula to get how many copies will be there after each round. So I normally use this formula 2 to the power of n. So each round I represent as n. For example, after the first round, I will know I'm going to have 2. This first round, I'm going to have 2 to the power of 1. That's the first round. Then 2 to the power of 1, this means I'm going to have 2 copies, because if you do 2 to the power of 1 in the calculator, it will give you 2. So after round number 1, I'm going to have 2 copies, which is the example I just did now. That was the first round. We had 2 copies at the end. After the second round, again, same thing, same formula, 2 to the power of 2. And then again, if you put on the calculator, 2 to the power of 2, that will give us 4. So now, second round, I have 4 copies. Same will apply. Third round, it becomes 2 to the power of 3, which becomes 8 copies. Fourth round, 2 to the power of 4, 16 copies. Round number 5, 2 to the power of 5 becomes 32 copies. Round number 6, 2 to the power of 6 becomes 64, and so on and so on. So if in case they would ask you how many copies will be there after each round, this formula to me was quicker way to always get the answer fast. And then yeah. So also there are this type of questions where they're going to ask you to calculate the percentage of the nitrogenous bases which are there in your DNA molecule. For example, just thought of this one from my head now. Let's say I've got a DNA molecule which consists of 20% thymine. So we know that 20% means to raise 20 over 100. So I've got 20% which are thymine inside my DNA molecule. So we know by now that thymine always complement with adenine. So if I'm having 20% of thymine, this means I must have also 20% of adenine. 20% of thymine, I must have 20% of adenine, because they go together. So now I've got 20% of thymine, 20% of adenine. So I'm having 40% between A and T. So A and T is giving me 40% in this DNA molecule. So A and T is giving me 40%. That's 40%. So 40% is coming from A and T. So now because I'm having 40%, I know in my DNA molecule at the end it must be 100%. So these two are giving me 40%. Therefore, I'm left with how much? 60%. That is remaining. And again, this 60% we know it must be shared between guanine and also cytosine. And also guanine and cytosine, we know they also share the same amount. So this means guanine will get 30%, cytosine will get 30%. And the question I was asking, what is the percentage of guanine and present? Meaning the answer will be 30%. Answer will be 30%. 30%. So that's it, guys. So I've seen sort of like these questions being asked in multiple choice when they're asking for different percentages and stuff. So last thing I want to cover today is the DNA fingerprinting. So mostly this is used by police officers because I normally watch a lot of shows where they are using DNA fingerprinting to identify an organism or like an individual who's guilty or something like that. So DNA fingerprinting, the method where they're identifying or comparing the DNA which is there was found before. So they use X-ray film, which normally gives like a black bars behind. So I found one picture online which I thought it was a nice example to use. For example, let's say in a crime scene, this is my crime scene. Say there was a building somewhere there, then they found a crime there, then they went and took the DNA, then they found this crime scene DNA as present, and then they found also, or they're suspecting 3 people to have been the one who did the crime. Then they go and take their DNA, and then they do the black bars. So you need to go and compare all 3 suspects with your crime scene. For example, here in my crime scene, there are 2 bars which are there at the top. This one, number 1, does not have the one at the top, but then there's one at the bottom. Okay. Here the black bar is down there. Here is down there. Here and there's no there. So this one in the crime scene is only one big. Here there's 2 of them. So automatically you can see that one and crime scene are not linking to each other. This one, number 2, look here at the beginning, 2, same. So number 2 looks similar, like in 98% or looks so similar to the crime scene. Number 3, okay, there's one at the bottom, there's 2, there's one big. Okay, no, it's not the same pattern. This pattern is linking to number 2. So we must be careful now. We can't really say number 2 is the one responsible for the crime. Their friend, his or her friend, could have went to the crime scene and put the DNA there, or someone else could have put the DNA there. But one thing you can conclude is that there's a big chance, or the DNA of number 2 was there in the crime scene. How come, that's a different story, but you can't conclude now and say number 2 is guilty, because the DNA could have been by someone, or someone could have used the number 2's jacket, something like that. But so far we can conclude on this, or number 2 is more linked to the crime scene, but we can't say number 2 is guilty of whatever crime is being investigated here. So again, the uses of DNA fingerprinting, besides the one we just mentioned now about crime scene, it can also be used for diagnosis of disorders such as hemophilia, which I'm gonna do also when we do meiosis, and also sex-linked disorders later on. So also it can be used for developing cues, this DNA fingerprinting, also for finding relatives, since we're using the DNA. Remember from the last lesson that DNA is important. That's why genetics is very crucial, because you can track families using DNA or genetics. So last thing, also, everything that is good tends to have sort of like the side effect, or people who will oppose it. Some of the examples we're going to do is when we're doing cloning and also genetic modification. There are people who are also against those ones. Even this one, DNA fingerprinting, people believe it's controversial because we're using a small portion of DNA. So people believe that this DNA might not be unique to the individual whom we're investigating, for example. Also, some private labs, since now I can just go and open my own private, there's a big chance I might not follow what is required because it's my own. So people always believe that private ones can just go and do as they please. And also, people are human beings, so a human error is possible. An error could have happened. Someone is doing this experiment, they're tired, they haven't slept for days. Like, a lot of things can go wrong. And also, it's very expensive. In South Africa, most of us are from poor backgrounds, and most of us, to be honest, we are poor. So it's very expensive to accommodate everyone. I can be accused of a crime, and I can't back myself up because it's too expensive for me to do it. And lastly, it can also reveal some diseases which are in my body, which I don't want anyone to know. Thank you very much, guys.